This is TWIS. This Week in Science, episode number 763, recorded on Wednesday, March 4th, 2020. March 4th with science. I'm Dr. Kiki, and tonight we will fill your head with bangs, birth, and death. But first, thanks to the Burroughs Welcome Fund and our Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience. Disclaimer, disclaimer, disclaimer! This Week in Science is coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Oh, and I'm waiting for Justin to say good science, everyone. And, you know, Justin's not here. It is a different kind of show tonight. That is right. Happy Day of Science. Welcome to This Week in Science. Got a great show for you. On tonight's show, we have new hosts. Surprise!
What?
Yeah, what? What is going on? Blair and Justin are both gallivanting around the world. Doop, 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 doop. They're off on their various adventures. And so I get to hang out. I feel extremely lucky to have two doctors on the show. This is the Triple Madness Doctor Science show. I'm joined by Ethan Seigel. He's an astrophysicist. You may have seen him on the show previously. Welcome to the show.
Thank you for having me here. It's a pleasure to be here. We're going to have some amazing science this week.
I think so. And our other guest co-host tonight is Dr. Jessica Hebert. She is a placentologist and has also previously been on the show. Welcome. Thank you for joining us.
Thank you for having me. This is my third time on TWIS, because my first time I was a scientist and the second time was at the live show at Alberta Rose when I performed with my band, the PDX Broadsides. So it's good to be back. Hi, TWIS.
Hi. I'm so excited to dig into science with both of you, and I know everyone is ready, chomping at the bit for some good science stories. So tonight I brought stories about a lot of things to do with, a coronavirus you might have heard of us talking about, a little bit of gamete chromosomal influence on some other things, and additionally, was Earth a water planet at one time? Maybe. Ethan and Jessica have both brought stories because they are the guest hosts as well. So, Ethan, what did you bring for this show tonight?
Well, it was very exciting last week, at the end of last week, so you couldn't have covered it on last week's This Week in Science. The NASA Chandra X-ray Observatory announced a discovery of the largest explosion in the history of the universe. And I am excited to tell you about that. It's not a supernova, not a hypernova, not even the merger of two black holes, something else that was even more energetic. And so beyond that, I'm going to tell you what the biggest explosion that we know must have occurred in the universe since the Big Bang is. And that one we haven't found yet, but someday we're going to. And that's what I've got for you this week on This Week in Science.
I'm super excited. Okay. An explosion that hasn't happened, but we know it has. We want to find it. All right. Jessica, what did you bring?
Well, I've got an expose on the powerhouse of the cell. What secret, exciting dance has it been hiding from us? Also, a whale of a tale. This coming up on This Week in Science.
Woohoo! Whales, tails, large explosions. We've got so much fun coming up. So let's get into it. But before we do, I would like to remind everyone that you can subscribe to the TWIS podcast on your favorite podcasting platform, YouTube or Facebook. And TWIS will bring you new episodes every single week. Well, whenever we publish new episodes, really. Just search for This Week in Science or visit twis.org. But now it's time for the story. So let's get to the COVID update. COVID-19 is the name of the disease that we have come to know, and some people fear. And it is caused by the virus that has been renamed SARS-CoV-2. And this virus has made a wondrous spread from China, its origination in Wuhan, possibly due to bats, potentially pangolins, definitely not snakes. We know that now. Snakes were not involved. And this virus has now made the trek around the world, is reaching more and more countries. We are now over 95,000 cases that have been diagnosed, over 3,200 deaths as a result of the disease, but a large number also recovered. Over 53,000 people recovered from the disease. We have had 9 deaths in King County, Washington. Additionally, researchers in King County, Washington are also looking into what is happening with the spread of SARS-CoV, and they've been looking at genetic sequences of SARS-CoV. There is an open database called NextStrain that is available for anyone who wants, and it is created on GitHub. And so while right now they are looking for a couple of full stack developers to help them on this project as paid jobs to study the genome analyses of COVID-19, they're also looking for volunteers to help maintain and update the data as it comes in. The genomic situation, we have discovered that it is developing mutations as it travels through different populations in the United States. It was introduced into Italy at least twice, with subsequent community spread, and most recent studies of the phylogeny, the analyses of these genes and the mutations, have determined that there are possibly 3 or more strains that are in circulation in Washington state currently, and that they have probably been there for about 6 weeks. So potentially we're about 3 weeks out from a big bada boom in Washington with respect to this virus. We don't know. But if you have questions, if you have concerns, there are wonderful resources on the internet. We will post a link to this NextStrain database so that you can peruse it. And you can also email us here at This Week in Science, Kirsten at thisweekinscience.com. Now, Ethan and Jessica, what kind of concerns do you both have about not just coronavirus, but COVID-19 specifically? Thoughts?
One of my concerns is what's called the R naught on COVID-19. So the R naught is so I'm infected, how many other people will I infect? So measles has an R naught of it's big. It's like 15, 17. So that means for every person who's infected, they can infect 17 other people. The R naught on COVID-19 is currently sitting about 2.5, which is a little bit more than SARS was, or a little bit less than SARS and a little bit more than MERS. So that's kind of nerve-wracking to know that it can spread pretty rapidly, and also that it's sort of a sleeper. It seems to be carried by a lot of people who don't necessarily have symptomology. So what does that mean for the long-term sort of incubation period? Are people going to be incubators for a long time and just not know? What does that mean for at-risk populations? Someone becomes a carrier, doesn't know, and they're a nurse at a nursing home. That's a very vulnerable population. I feel comforted that the young are not being affected. And they are, but they're not the population that's dying. So that's That's good. And unlike Spanish influenza, it's not killing healthy middle-aged people. So I think I'm safe, at least a little longer. Wash your hands like your Lady Macbeth, though. Like, get that blood off your hands.
Out, damn spot.
The DuckTales chorus, sung twice, is about 20 seconds. So is the chorus of All Stars, sung twice. Just like scrub, sing loudly to people. My lab found out that 2 courses of DuckTales is what'll do it when I started singing it very loudly yesterday. So
you got to get through it any way you can. And not everybody wants to sing the alphabet or the happy birthday song.
Yeah. So please take care of yourselves, wash your hands, touch your face as little as possible, and just stay healthy out there. You do not need to be wearing a mask if you are not sick. Stop mass buying them.
My big concern about COVID-19, and this echoes something that Jessica alluded to, is that when we typically have something like the flu, a cold, one of these more common illnesses, we say, okay, like from the moment you're symptomatic, you don't go out in the world. You don't go out and you don't go and infect other people. You stay in your house until all the things go away, until your fever breaks, until you are not like blowing your nose all the time anymore, and you're less likely to transmit disease. The problem that we are seeing with COVID-19 that's different from the flu, from a cold, from things like that is the incubation period. That's the time from when you contract it and when you can pass it on to others until you start showing symptoms. And as far as I'm concerned, what I've been reading, and you can correct me if I'm wrong about this, is the tests that we have to test for this will not necessarily give you a true positive if you are positive for it until enough of this time has passed. That incubation period can be a long time. Typically, you think like, a few days at most. It actually, for COVID-19, I've been reading that the incubation period, it can be as little as 2 days, but it can be as much as 27 days, which means you can be hanging on to the coronavirus. Like I could have, it's not me, but it could be, right? It could be anyone, because you can have no symptoms for up to almost 4 weeks and be infecting everyone you come in contact with, because it's an airborne virus. And so I think one of the things you need to be careful about is, yes, wash your hands a lot. That's a great way to do it. If you're going to use hand sanitizer, use hand sanitizer for real. Stop touching your face. This is a great opportunity to get that terrible habit. Don't touch your mouth. Don't touch your nose. Don't touch your eyes. Try not to touch your mustache. I've been ruining with that one,
How will I survive?
I just wonder.
I don't know. It's like the old Hollywood classic is, when you're alone in your house and you're getting ready, you groom yourself to make sure you look perfect. As soon as you walk out, never look again, never touch it again. Just don't worry about it. Just go out, know you've nailed it, and don't ever doubt yourself. Don't look in a mirror.
Don't look in a mirror. Just keep going.
But that's what concerns me, is that there are a lot of people who could be carrying it, who look asymptomatic. And that doesn't mean go out and be paranoid, but it does mean be mindful, because people you run into, you might not know they have it. They might not know they have it. They might have it. And so I would say, just in the back of your head, don't go doing your life any differently than you normally would, with those few exceptions about be good about hand washing. Be good, of course, if you have to cough or sneeze, into your elbow is ideal, right? Not into your hands and then touch other people. So these are like basic public health advices, but also do be mindful that this is out there. If you get it, it's probably not the end of the world, but it does have a significantly higher death rate than, say, standard influenza, which has already killed 10,000 people this year. So we're talking 2 or 3% morbidity if you get it. My more realistic concern is that coronavirus, COVID-19 in particular, is going to become, and we don't know if this is true or not, that it might become just a seasonal thing. That, every year you might have to worry about not just getting the flu or the swine flu or the bird flu or whatever variant is out there and deadly this year, but but you might have COVID-19 on top of that. This might not be something we have a single vaccine that works against. Based on the flu rate vaccinations, I doubt most of the country or world is going to get that vaccination, even if it does exist. And this is just a reality. This is a public health problem, and this is something that it's likely going to be on our radar, certainly for the rest of the season. And we don't know if it's going to continue beyond when the warm weather comes to the Northern Hemisphere. We don't know if it's going to continue on and still be a threat like that. And we don't know if it's going to persist to be a worldwide problem in future years. This is something that is probably not going away anytime soon.
Yeah. And, if we do get a vaccine that is one more tool in the arsenal, if it does become something that is seasonal or even just is around all the time, we don't know if it's going to go away in the summer with heat. We have no idea. Someone in the chat room is asking, does your temperature at least go up above normal a bit when you get it? And during that incubation period, no, you don't show. It's asymptomatic, but it's at the point where you start to show symptoms. Usually the first symptoms are a cough, a high fever. Your fever goes up to 102, 103 degrees. And there are other aspects in there. Difficulty breathing because it's a respiratory virus. But the fact that it is, as you said, airborne, it's not airborne. It's not like I'm breathing and there's just virus going out into the air. It's droplet based as far as they can tell. So it'll go into the air in droplets of saliva or snot and mucus. And those droplets then stay in the air for a little bit, but end up landing on surfaces. And so the big deal is those surfaces. And that's the reason we touch things. And that's the reason we really need to be washing our hands, because someone sneezes, I rub my nose, and then I touch a computer or do something like that. And then you come along and touch the computer or whatever, and it gets transferred.
Yeah, and 9 days, people. 9 days is the amount of time that if I hock a loogie on something and Dr. Kiki is unlucky enough to touch where I hocked, 9 days later, she can still get COVID-19.
Unless she washes her hands
and cleans the surfaces and
before she touches her face.
Right. Your hands, your skin is an excellent immunological barrier. So washing your hands before you touch a mucous membrane is going to help protect you. And if, like me, you're planning on traveling, I'm flying to the Society for Reproductive Investigation conference, all the placentas you can shake a stick at on Friday.
Shake a placenta at.
Shake a placenta. There's a lot of concerns about what can you do to keep yourself safe during travel? Should you wear a mask? Unless you're sick, don't wear a mask. It doesn't do you any good. Unless you're really immunocompromised, then, it's going to protect you against other things. And mostly what it's going to do is keep moisture in your face, and drying out in this area is what really makes you susceptible to some of these droplet-based spittle projectiles in the air. So keep yourself moisturized, wash your hands. And if your local store is out of hand sanitizer, you can mix isopropyl alcohol. So 99% isopropyl alcohol with aloe vera gel. Yes. If you want, you can pop a couple drops of essential oil in there, shake it up. It needs to be 2 to 3 parts isopropyl to 1 part aloe vera, because you need to have at least 60% alcohol in order to kill what's on your hands. If your store is out of isopropyl alcohol, find 99% Everclear in your liquor store. People have probably not raided the liquor store yet. It's fine. And you can also mix it with orange juice and have a cocktail. So it works really well.
A little bit with the orange juice. Put more with the aloe vera gel.
Allegedly.
Yeah.
Allegedly.
You can choose your own adventure on that front. There's no public health advice against that.
You can also take a little Ziploc baggie with Clorox wipes, wipe down the surfaces before you sit in that plane seat. Just take the normal precautions, take good care of yourself. And if you feel like you're getting sick, smack a mask on your face and go home.
Go home. Stay home. Yes. don't travel while sick. Don't go to work while sick if you can avoid it. And
There's a question about whether vodka is strong enough.
Not vodka, no.
So what you can do, if someone distinctly legally not me were to tell you that you could use dry ice to freeze out some of the water, you could increase the alcohol concentration of vodka, and that might work. But it's just easier to buy isopropyl alcohol and far more legal.
Far more legal. And
yeah,
don't drink it. Just use it for your hands.
Yeah.
But if you do think Everclear is gross and you do want something to drink, I can recommend Winemaker's Brandy, which is 190 proof. So enjoy.
Well, that's how you sterilize your insides for science.
For science. Oh, sterilization. That is something that the air should be doing. So researchers looking at scans of the skies of the atmosphere over China have discovered that over China, air pollution has dropped dramatically since they've been dealing with COVID-19. So since COVID-19 has been around, they have seen a massive drop in pollutants over the country, and it can be directly linked to their manufacturing plants being turned off because people have.
It's like a Billy Joel song. It's great. They're closing all the factories down.
Oh.
So while China comes boots back up and starts manufacturing again, this is going to be very short-lived, but it is a very visible sign of what we are doing to the air with the burning of fossil fuels and with the manufacturing that we do. We put lots of stuff into the skies, and they do have an effect. Another study just out this week, published in Cardiovascular Research, looked at a data-informed atmospheric model to calculate worldwide exposure to particulate matter and ozone pollution. So not just particulate matter, but fine particulate matter. This was combined with estimates of disease-specific excess mortality and loss of life expectancy from the year 2015. And they looked at lots of different pollution sources, whether they were natural or human-caused, anthropogenic, including fossil fuel use. And they discovered that air pollution from human activities, from air pollution from all activities, all causes, natural or human, but human is the one that we can really control, that air pollution is a bigger factor at reducing life expectancy globally than tobacco smoking, malaria, and many other diseases. So if we could just, figure out how to not pollute so much, people could potentially live longer and healthier. And if they weren't living with cardiovascular and respiratory illnesses or problems that are caused by air pollution, potentially they wouldn't be in a situation where they would succumb to things like COVID-19 so easily. It all comes full circle, people.
When you look at the leading causes of death worldwide, right, we get things like cancer, we get things like cardiovascular disease like heart disease and stroke. And then competing with that for the top 3 spots worldwide, we have things like COPD, which are directly related to things like air quality. And one of the concerns I have where we all live here in the Pacific Northwest of the United States is wildfires have been on the rise in the summer months.
Mm-hmm.
And we've had days where it's not really safe to go outside because of the air quality. And I'm kind of wondering, are we at risk of this? Just like, we know that people who heat their homes using wood or other biomatter that produces a lot of like particulates, they are at a much higher risk of getting COPD. People who live in heavily air polluted regions of the world, places like Calcutta, India, they're seeing like enormous rates of COPD as the cause of death. Is this something we're going to have to start worrying about here on the West Coast as our air quality gets very poor certain times during the dry, hot summer months where there isn't as much water as there used to be?
Yes, that is, it is definitely because this particulate matter, the smoke that enters the air from these wildfires, you have the air health warnings for a reason. There are certain days where it's recommended that you just don't go outside because it is dangerous. And exposure for long periods of time can be detrimental to your health. Absolutely. So it's not on the matter on, you're not cooking inside a small room with no windows with a wood stove, but at the same time, if you're surrounded by this and you're breathing it, it's going to have effects. There have been other studies looking at levels of various diseases in people who work at toll booths on freeways, and they find that there are higher levels of cardiopulmonary disease, of cancer, of all sorts of disorders in people who work by where cars come through all day long. And it's thought to be because of the exhaust from all the vehicles driving through. Yeah. And they, over the period of the Chinese Olympics, they shut down all of the manufacturing right before the Olympics and during the Olympics because the air quality was so bad. Many of the athletes were saying, we don't want to go. This is not going to be healthy for us. I can't compete and breathe. My body is a machine. I can't put this machine, this fine-tuned race car machine, into this bad environment. So they cleaned up the air for a bit and they looked at pregnant women who had the third trimester of their pregnancy during that period of reduced smog in Beijing. And they found that those women were more likely to carry their babies to full term, and the babies had higher birth weights.
That's fascinating.
Yeah.
I absolutely want to read that paper, being someone who studies pregnancy.
Exactly.
Yeah. There's a field called the Developmental Origins of Health and Disease, or DOHAD, which was born here in Portland at Oregon Health and Science University. Yeah. But it's what happens in utero that affects you for the rest of your life, and then potentially affects future generations. So there's been study like the Dutch famine crisis, pollutants, like what happens over generations and what happens at different trimesters, at different developmental points that affect you differently. If you have poor nutrition in the first trimester, is that better or worse than having poor nutrition in your third trimester?
Yeah.
And there are differences. And there's differences between boys and girls too. So boys are terrible to their placentas. Just the worst.
Why? Why are boys so bad to placentas?
How much time you got?
I know. Oh boy.
One of my favorite papers in the field, I'll keep it brief. It's called Boys Live Dangerously in the Womb. And just go take a look. It's not very long and gives you kind of a brief overview of why are boys terrible to their placentas.
All right. I think I've done all the depressing stuff for the show now, so I'm done with it. I decided, we'll just rip the Band-Aid off, get it all done with at the very start. But I'm going to be done for a while. Ethan, can you tell me a story?
I can tell you a story. So I assume you want to know the story about, people were reporting this as the biggest bang since the Big Bang. And when you think about like things that go bang in the night or the day, because, the universe is 360 in all directions, we don't discriminate in astronomy. We just look where we can see, which is at the night, usually from the ground, but from space, we can look anywhere we want. Typically, you think of things that go bang and you're like, oh, like stars can like have nuclear fusion happen. They can like go nova. They can have their cores collapse and go supernova. Those are really energetic events. When a star goes supernova for some time, for maybe even almost a year, it can be about as luminous as like 10 billion normal stars, which is about 10% as bright as a typical entire galaxy, except concentrated into one tiny point. And you say, how much energy is that? And basically, if you took all the energy that the sun outputs over its entire life, 10 billion years, 12 billion years, and said, I want to emit this all at once. I want to emit this all in 1 year. That's how bright a supernova is. It's basically like if you took the planet Jupiter, right, biggest planet in our solar system, and said, hey, I learned somewhere about Einstein's equation that E equals mc squared, and you just took all the mass of Jupiter and you multiplied it by the speed of light squared, and that's a big number, right? Speed of light, pretty fast. Speed of light squared, pretty big number. Multiply it by the mass of Jupiter, pretty big, about 1,000 times as massive as Earth, right? That's how much energy a supernova emits all at once. We're talking about 10 to the 44 joules or so, and that's not even close. That's like 1 billionth of the biggest bang in the universe. So you go, all right, what's bigger? What's bigger than a supernova? And you say, well, how about a hypernova, right? How about a superluminous supernova, right? You're on the right track. You're 100 times bigger now. Good job. Still not close. Not even close. You're not even comparable. Why not? Well, because you can turn more matter or whatever into energy. You can do something bigger. So you say, aha, I got it. What about when LIGO found those merging black holes and they turn literal, like multiple solar masses worth of material into pure energy in the form of gravitational radiation? And you're right, that's bigger. That's more energetic than even a superluminous supernova. But you're still down more than a factor of 1,000,000 from the biggest things in the universe. Believe it or not, the biggest thing we found just last week, it was announced, comes from a giant cluster of galaxies. So forget about single stars, forget about multiple stars, even forget about like individual galaxies like the Milky Way, which are collections of like 400 billion stars. We're talking clusters of galaxies, which is like thousands of Milky Way style galaxies all in one group together. And what we've got up here, this is an image, it's a composite image of what we know as the Ophiuchus cluster. And this is just, Ophiuchus is a constellation. This is a galaxy cluster in that constellation. It's about 390 million light years away. And you see that big white spot in the middle of the pink. That is one of the brightest, biggest galaxies in the whole cluster. We know it has a supermassive black hole at the center of it that is much bigger than the supermassive black hole at the center of the Milky Way. We're talking about 1,000 times as massive, which means its event horizon is like 1,000 times as big as the one at the center of our galaxy. And what happens when you get these supermassive black holes? Every once in a while, this matter falls into it. Maybe you had a merging galaxy cluster. Maybe you had like all sorts of things. Maybe you had two galaxies merged together. What happens is a large amount of matter falls onto that black hole. It gets accelerated and it emits right there. It emits jets in two directions, right? You see it's coming out in two mutually anti-parallel directions perpendicular to the plane of a disk galaxy. These jets can be super energetic. They can last for a super long time. And they're basically accelerating particles to speeds that are indistinguishable from the speed of light. They're going so fast. And what they do in this galaxy cluster is these jets slam into all of this gas and dust and plasma in the intergalactic medium, in the intracluster medium, the space between the galaxies. So this matter goes, it hits it, it makes this enormous cavity. So here you see a supermassive black hole. This is one, we're looking at it in three different views. This galaxy in the top view, that's Messier 87. That's the biggest galaxy within about 50 or 60 million light years of Earth. It has a black hole that's 6.5 billion solar masses. And you can see up top in the optical, down below on the left in the radio, and down below on the right in the X-ray, you can see it is emitting jets in those two anti-parallel directions. The big one that's coming off, it looks like it's coming off to the upper right of the image. I can't make my hand go upper right because I'm all flipped in my camera. But you go and do this, and what happens? This jet itself is thousands of light years long. And it's not even that spectacular. Why is it not even that spectacular? Because it's not even that energetic. The one we found, right? I said that's a few thousand light years long.
Which
The
seems
one we
huge.
found is about 15 times the size of the Milky Way. That blue light you see, that's radio emission. That red light you see, that's X-ray emission. And that line, that dotted line you see that says cavity edge, that's what we're talking about. This thing carved a cavity in space that's about 1.5 billion light years in diameter.
Yikes.
So you go and say, oh my God, what happened? What could cause that? And the answer is, it must have been from someplace else, a jet that existed for a long time, expelled a ton of energy. And by a ton, I mean, it's many, 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 teratons of energy. We're not talking tons like metric tons. We're talking like, this is about the equivalent of about 10 billion normal supernovae. That's how much energy we're talking about. It carved a cavity that if I were to line up Milky Way galaxies across it, I'd need about 15 of them to go from end to end. That's how big of a cavity it blew into there. If you want a number in terms of joules, it's about 5 times 10 to the 54 joules of energy. So you say, wow, that's impressive. How much bigger is this than anything else we've ever seen? Well, the answer is, yeah, we've seen supernovae, we've seen merging black holes. As far as cavities in the gas in giant clusters of galaxy goes, this one's about 10 times bigger than anyone else we've ever seen. This we can safely say of everything we've ever found directly in the universe, this is the largest single event in terms of energy ejection that we've ever seen, and it's not even close. But what I love about this is this Ophiuchus cluster is only 390 million light years away. We're seeing it with the relatively primitive, and I'm sorry, because it is the greatest X-ray observatory of all time, but it's 20 years old now, and it's time to go to the next generation of stuff. So this coming decade, here in the 2020s, the European Space Agency is going to launch the Athena satellite, which is like hundreds of times better than the Chandra X-ray telescope in a large number of metrics. The next decade, in the 2030s, there is a large group of people trying to get this mission called LYNX, this NASA mission called LYNX, which would be even better than Athena, even better resolution than Chandra, to launch that into space. We would, no doubt, be able to see events like this that aren't just, 390 million light years away, because that's close by. The universe goes out like 100 times that distance in all directions. With next generation telescopes in the X-ray, like ESA's Athena, like the proposed NASA LYNX mission, we could find these giant energy ejections events possibly anywhere in the universe. And that's one of the things that excites me the most, is this thing that we found. This is the biggest energy injection, the biggest, what some would call an explosion, that we've ever found in the universe. But we have every reason to suspect that there are even bigger ones out there. This is not the most massive black hole we've ever seen. This is not the biggest galaxy cluster we've ever seen. It's not the highest temperature gas. It's not the densest collection of galaxies. It's just kind of a typical big galaxy cluster, which is sort of like going to your local gym and finding like the biggest guy in the gym and being like, that's the strongest man in the world. No, it's probably not. It's just the biggest dude in your small pond gym. You want to go find the biggest energy ejection event in the universe? You better go out and look at the whole universe. And that's what I'm excited for, because it's not just enough to say, here's the biggest thing we found so far. I want us to go out there and I want us to see it all.
Yeah, I want to see it all also. So this, to me, I am really like struck with the parallel of geologists looking back through time and finding evidence for like super volcano eruptions and being able to say or looking back and saying because we see such such a scattering of a particular mineral or a particular particulate matter at a particular sediment around the world, we know that such a meteorite impact took place. Like, is that with this cavity that you're seeing and the light that you're seeing here? Is it kind of like forensics in
It's
space?
absolutely forensics, right? In fact, there were some people who used terms in astronomy. See, I just touched my nose. Trying to not do it. But yeah, there are people who use terms in astronomy like galactic archaeology, because what we're basically doing is we have this big problem in the universe that when we look out, we only get a snapshot. We don't get to do experiments the way that like biologists like Jessica get to do experiments, which is to say the laboratory is not an experimental one. We can't like say, all right, I'm going to take that black hole, I'm going to feed it a bunch of matter, I'm going to watch it make a jet, and then I'm going to see what happens. No. This is an observational science, not an experimental science. For astronomy, you can only look at the laboratory that the universe gives us. Now, it gives us a really big laboratory, right? We have a universe that has an estimated 2 trillion galaxies in it that we could ever possibly observe. That's a pretty good deal. That's a lot of things to look at. So you're going to find a lot of things. But in this one particular instance, you only get it in a snapshot. You only get it at a moment in time.
Yeah.
We know that quasars and active galactic nuclei and other black holes, they turn on and they turn off, depending on whether they're actively feeding or not. And they leave these signatures. What we're looking at here, this is a very late stage example. After the jet has turned off, after the energy ejection is complete, and we're just looking at the aftermath. It is kind of like looking at a crater, except in this case, I think we're looking at more like a bubble. It's like a 3D blown bubble in this plasma. So that's what we're seeing, is we're seeing what could have done this, what could have carved this bubble there. We've seen smaller scale examples of this, and we've seen things evolve on short time scales in like individual star clusters that have that type of energy injection. But we have to look and reconstruct and say, okay, we see this bubble in there, we have this idea of what processes are at play, so we know what could have caused it. How much energy would it have to inject to do this, and what could have caused that? And it does. It's like a detective story where you've got 4 different pieces of evidence all pointing to it, but you have to be the one to draw the conclusion. You can't exactly interrogate the witness and get a confession, right? You can't Vincent D'Onofrio this. You have to go all the way and say like, well, we're going to draw the best conclusion we have with the evidence we got. But there's this problem, right, that we can never really overcome in the universe, is you can never see what happened eons ago. That's already erased. That's already been erased by the universe. When it comes to what we see today, the universe only gives us the survivors to examine. And we have to use what we see of the survivors to reconstruct what happened thousands or millions or even billions of years ago.
That's the thing that interests me the most, I think, about astronomy, is what you observe isn't happening in real time. It's already done. So,
the stuff we're seeing right now, this is already 390 million years old from right now, because it took that long, maybe a little longer actually, for that light to travel to our eyes. And that's it. Like, so we even if we call that right now, right, what we're seeing is still the aftermath of the explosion. If that explosion were happening right now today, first off, it would last a while. It would likely last at least hundreds of thousands, possibly even up to like tens of millions of years.
Wow.
So it's not being emitted all at once. It's being emitted a lot of energy over time. But then you look at it and you say, well, if that were happening now, and we see jets in space. There's a beautiful galaxy called Pictor A that you can all look up and see like, oh man, now that is a jet. But
I'm not going to say that. No.
Oh, you will. You will when you see it. You will. You'll say like, I wasn't going to say now that is a jet, but then
will
you'll
I
see
sound
it. You'll be
like
Damn.
Like a farmer out in
Damn
the
it
field,
is.
like patting my John Deere going, that's a tractor. I'm going to be, that's a jet.
That's a jet.
Good. You know what?
right.
I will take that bet about the jet.
I'll tell you what. The next shot of Everclear is on me. If you find Pictoray, that is a jet.
I don't want your Everclear. I'm good.
Click on the blue one, the fourth image, the one that doesn't have those X-ray radio lobes over it.
Oh, that's a jet.
Chandra. That's a jet.
That is a jet.
Loading right now. There
See,
we
it's
go.
even from the same telescope, from the Chandra X-ray telescope. Look at that. That's a workhorse observatory right there. You know.
Okay, so I have a question though. So these jets, you really don't want to be in the line of fire of one of these jets, right?
You
all
say
this energy
as
coming
though
out.
Would we? You say this as though we aren't.
I'm sure it's from all around.
We are.
Yeah,
some of these jets, we are. Depending on your perspective, we're either super lucky or super unlucky that these jets are pointing right at us. So we see these quasars and these active galactic nuclei all throughout the universe, and some of them are oriented right at us. And when we see them, we originally gave them a different name. We called those events blazars because they're blazing right towards you. But guess what? They're the same object. Quasars, active galactic nuclei, blazars, they're all the same thing. The only difference is the orientation they have with respect to you. Blazars, in fact, give us the origin of the most distant cosmic particles we've ever seen. The Ice Cube collaboration detected a particle a couple of years ago from one of these blazar jets. We were able to identify it. We were like, oh, wow, that's not only outside the Milky Way, that's millions and millions of light years away, maybe even billions of light years away. And guess what? Comes right at us. So far, most energetic single particle we've ever seen, most distant identifiable single particle we've ever seen from a blazar.
All right. So not necessarily bad, depends on your perspective, to be in the line of fire from one of these jets.
Yeah, just don't get too close because it will roast you super good.
And if you're looking for a roller derby name, Blazar is pretty good.
That would be good.
Yeah.
Yes. Okay, let's move on from these West Side stories of astrophysics. And I'm going to move on into some biology. Dr. Hebert, what do you have for us?
Well, I'm going to talk about my favorite little organelle. When I say mitochondria, what comes to mind for you?
Powerhouse. He was talking about the strong man in the gym. I'm thinking of that little strong man in my muscle cells.
It's the thing that we all remember from middle school biology, right? Mitochondria, powerhouse of the cell.
Yeah.
Well, as power plants and energy stores, mitochondria are really essential components in almost all cells, plants, fungi, and animals. And until now, it's been assumed that those functions are because of a static structure in mitochondria, that nothing is really moving in those membranes. But researchers at the Heinrich Heine University Dusseldorf and the University of California, Los Angeles, supported also by the Center for Advanced Imaging, have now discovered that the inner membranes of mitochondria are by no means static, but rather they change their structure every few seconds. And it's this dynamic adaptation that increases their performance and allows them to be more adaptable and generate ATP. So in this, in our opinion, this finding fundamentally changes the way our cellular power plants work and will probably change the textbooks, says Professor Dr. Andreas Reichert. And I think so too. It's the fact that we are now talking about the dance of the membranes. So the membranes of the mitochondria move, which increase the amount of ATP they can produce at any given time. And if you'd like to know more about this paper, you can read about it in EMBO Reports. It just came out.
EMBO, and it's a whole big paper there. It's available online.
Yep. And it's open access right now. So do get at it.
Yeah. So what is it about the movement? Why would being able to move and change the structure, why would that make any difference?
So
I think this is fascinating.
I don't know why, and I don't think that they do either. But what I would speculate is that the mitochondria have different layers. There's four main layers to your mitochondria. You have the outer membrane, the inner membrane, and then the cristae and the matrix. So inside the matrix is not where you will find Keanu Reeves, but rather the complex mechanism of the electron transport chain that allows, when you've got your breakdown product of glycolysis, of fatty acid oxidation, of amino acid breakdown, all goes into here. You get those products and they're able to oxidize them to eventually drive what's called a proton motive force. So it's these little hydrogens that drive along and they turn the wheel of ATP synthase. And ATP synthase joins ADP with a free phosphate to make ATP. And the more you drive this, the more ATP you can make. Good deal, right?
So you need more electrons, more protons, more phosphate, more ATP.
That's right.
Pump, 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 pump.
But too much is also a bad thing. If you get too much, you get too much proton leak, and that can cause the formation of reactive oxygen species, which lead to oxidative stress and damage to the cell. So there's a balance in here. And I think that if you're able to move those mitochondrial membranes, maybe you're able to modify that proton motive force a little bit. If you're receiving, it might help you reduce oxidative stress. That's a theory that I'm spitballing right now. But if they end up using it in Dusseldorf, I would like grant money, please.
That's right. Pay up and let me know.
Yeah, mitochondria can move. They're doing a dance on their insides, which we never knew before.
See, mitochondria, they're like the most badass organelle that I know of, because as far as I know, they got two things going for them that no other organelles had. One of them is they have their own DNA. Like the cell has DNA, but I have my own DNA. I could just make more of me, whatever. Don't need that. The other thing, apparently, you can take mitochondria out of cell, put them in just their own environment, and they'll survive and thrive and reproduce like it's nothing. Like, what's up with that? Does that mean that, like, mitochondria used to be like, oh yeah, I was my own organism one day, and then like some other organism came on and was like, I'm going to learn how to make eat your DNA. Here we go. I'm just going to make you. You're a tool in my arsenal now.
Welcome to a great big debate in biology about where did mitochondria come from initially. Were they essentially phagocytosed into a cell, and when a cell said, hey, I could use energy. You're making that and you're kind of defenseless. Mitochondria are double membrane organelles, so they do have that sort of protective barrier already. But maybe it was a more advantageous symbiotic relationship in order to become part of a larger cell. The other thing that you mentioned about the DNA of the mitochondria, this is neat. It's matrilineal. You can look at mitochondrial inheritance and it goes up the mom's side. You get your mitochondria from your mama.
Oh, no Y chromosomes in my mitochondria.
It's more complicated than that. It's just you inherit your mitochondria from your mama and your grandmama and your great-great-great-great-great-grandmama. Like the mitochondria pass on down.
Ooh, yeah, that's kind of exciting.
It is neat.
Thank you, great grandma Esther. Bam.
She didn't just give you that bald head. She also gave you
Mama's mitochondria.
This light is extra shiny.
Male pattern baldness also passes down through the mother's line. Sorry.
Oh, good news. I have some fun news for the second half of the show related to gametes and inheritance and what that means. We'll get to that.
Exciting.
Yeah, I know you have one more story.
Yeah, would you like to hear it?
I want to say very quickly before you jump into it, though, that if people just tuned in, they are watching or listening to This Week in Science. And if you are interested in a TWIS shirt or mug or other item of TWIS merchandise, head to twis.org and click on the Zazzle link to browse our store. All right, go for it.
So I would be remiss if I came on TWIS and did not talk about Team Placenta. This is a placenta story, and because Blair's not here, we got to represent some zoology. For the first time in history, whale researchers have captured video footage of a humpback whale giving birth. On that day, they were also able to spot a floating placenta from a different birth in the same waters and collect it for histology.
Wait, what?
Yeah!
Floating placentas.
So you
Floating
have a
placenta.
recently birthed whale placenta, recently afterbirthed whale placenta from a humpback whale from a different birth that now, like, scientists can analyze. What normally happens to these placentae? Do they just like get devoured by some other whale, or does it sink to the bottom of the ocean and the sharks get it? Like,
well, if somebody doesn't find it, it gets eaten or it gets degraded. I mean, it's got nutrients in it, so it can be, consumed. And especially if you're in a low nutrient environment and you're hungry, I mean, that whale placenta is looking pretty good. If you're a human and you live within driving distance of a grocery store, don't eat your placenta. But,
the store.
Yeah, if you're a shark and there's a whale placenta, I mean, nom down on that. It's probably great. I'll pass, but it's nutritional. So this is kind of an odd treasure. You don't get a lot of humpback whale placentas. It's rare to find them, and they had never seen a humpback whale live birth today before. And Captain Steve aboard the Ocean Explorer, the captain and crew are no strangers to placenta collection. In 2018, the same crew collected a placenta under the direction of Pacific Whale Foundation researchers. In both instances, they reported the find to the foundation's chief biologist, Stephanie Stack, who called the fisheries to report the collection. Marine mammal parts are protected under the Marine Mammal Protection Act, which means collecting animal parts, even if discharged or sloughed, are not permitted unless you are operating under authorization. Yep, those are the pictures of the whale placenta. So you can see that it fills an entire, like, not your standard beer cooler, but like a huge cooler.
it looks like an inside-out octopus.
It does. That is complex.
It's like a placenta is an entire organ. It's the best temporary organ the body will ever make. And all of us make one. It's pretty, it has this very important job. It's to exchange nutrients and waste and gases between mom and baby. And if it's doing its job, it's going to look pretty gross. It's a lot of blood vessels, a lot of connective tissue, no muscle, no bone, a lot of immune cells. That's what it's there for. It's an exchange organ. A human placenta is generally 20% of birth weight, so it wouldn't surprise me if humpback whale is about the same. So it's going to be a big placenta.
Yeah. How many tons does a humpback whale weigh? I'm just 20%.
I'm not sure. Right. Let's ask.
20% of the weight of the birth weight of the baby, right? It's not, or of the calf. It's not 20% of the weight of the mother.
No, it's 20% of the baby.
Okay,
so a newborn humpback whale calf weighs approximately 1.5 tons.
Tons. Okay, yeah.
That's tons. And each baby whale consumes up to 52 gallons of milk per day. That's a lot of milk.
That's a lot of milk. It's a big appetite.
So yeah, we got to finally see a humpback whale give birth in the wild and then get the placenta from it. So we can ask all sorts of questions now about, like, so how is this placenta different than other marine mammal species? Is it similar or different to humans, to rodents, to other known placental mammals? And what does that mean for long-term sustainability of the organism? It's really cool.
So
it's still an N of 1, though, right?
It's an N of 1 for birth. It's not an N of 1 for placenta. I think this is the second that this crew has gotten, but it's definitely an unusual find.
Unusual. Okay. Go ahead, Ethan.
Yeah, I have this weird question, and maybe one of you two will know this because I'm not the bio person, but it's fascinating and weird and gross in a really in a way that I really like. I want to know more about. I read that in humans, human women who have been pregnant, will get some of the like healing powers of the baby's stem cells when it's growing inside of them, and they will become like chimeras where they will exhibit what's called chimeraism because they have some of these baby stem cells that have gotten into their bodies and can do things like repair organs and tissue damage and things that heal in no other way at no other time in any other humans. Like only pregnant women who've had like a child growing inside of them experience this. Do whales, do other mammals, do other non-mammals, basically does everything with a placenta exhibit this chimeraism? And if so, like,
No.
can I get some of that stem cell action? I got some broken parts I'd love to like just heal on up.
No, and what the placenta is releasing into mom's bloodstream is called microvesicles or exosomes. And they're little, think about exosomes, they're about 50 nanometers wide, and they're like the little emails being released by the placenta. And they do carry messages to mom, though what those messages are, we don't fully know yet. We do know that there is some remodeling of organs and blood vessels that can happen because of the messages enclosed in the exosomes. You can find RNA and DNA in mom's brain years after she's had a baby, but whether or not they're actually like healing or repairing her is sort of debatable.
Yeah.
Do all mammalian placentas do that? No. Not all mammalian placentas have that level of crosstalk and release exosomes, but some do. We know that mouse does, but there hasn't been a whole lot of research about it. Exosomes are still kind of that bleeding edge field, but we're learning more about them.
Okay, cool. Thank you.
Exosomes.
And there are, beyond the exosomes, I think there is fetal cell migration that does take place, but these are interlopers, and the mother's immune system does not like them.
No.
And the mother's immune system, once it has decided that it no longer has to protect the thing inside of the mother, because the placenta is done telling it that it has to, the mother's immune system says, attack, and will kill it off. And so these cells do not last very long.
There was a
as far as we
question
know.
in the chat box about what other temporary organs the human body ever makes. The placenta is the only one. That's why it's funny. Yeah.
And I love your comment, just to come circle all the way back to your very quick off-the-cuff comment that we all make one.
We do.
And that's because it's the baby that makes the placenta.
It's a fetal organ, not a maternal organ.
Yes.
Share that at your next cocktail party. You'll never be invited back. It's great.
Would you like to see where I grew a placenta?
This is why I don't have friends, Kiki.
Only the best of friends, because they appreciate.
It's true.
Appreciate it. It's true. Okay, we're going to take a break. I do love all this placental talk. It's rejuvenating in a sense. We are going to take a very quick break, and I mean very quick, for a moment, and we'll be back with some more This Week in Science. Stay tuned. Thank you for listening to This Week in Science. TWIS is here because of you. You're the reason that we're able to do everything that we do every single week, bringing you all the news with down-to-earth views. Well, maybe sometimes the views are out in space, but we do try to bring you the latest discoveries and pretty rational conversation about them, even though we enjoy the curiosity and the opinionology from time to time. With your help, we can do even more. Together, we can help bring this saner perspective to a world full of misinformation. You can help share TWIS and the perspective that we have, but also you can just head to twis.org right now, Click on the Patreon link and choose your level of support. Be a part of bringing sanity and science to more people. Thank you for being a part of TWIS. We really could not do this without you. And we're back with more This Week in Science. Now, I told you earlier that I had a gamete story. We weren't just going to let those chromosomes and conversations about maternal mitochondria go unfollowed. No. I have a story about death by gamete. That's right. Apparently, there is a hypothesis that has been studied, but there's a new paper out in Biology Letters this week about the unguarded X hypothesis. Now, the unguarded X hypothesis suggests that the Y chromosome, that's the chromosome in mammals that leads to being male, when you have an X and a Y, you become male. And if you have two X's, that's homogametic, is XX, you're female. So heterogametic, male, homogametic, female, XY, XX. Anyway, that Y chromosome is less able to protect an individual from bad, harmful genes on the X chromosome. And the Y chromosome, being smaller than the X chromosome, can't hide these harmful mutations that may be carried on a larger X chromosome. So it can expose an individual to poor health later in life. And so that leads to death earlier. Males die earlier than females. There's this thing. And we know that in humans, males have a lower life expectancy than females do. This has been seen in other mammalian species. Women just don't kind of have this problem. One healthy X chromosome can cover up for the problems of a harmful X chromosome. And so the researchers on this paper, they decided they needed to do a very wide range of animals comparing they need to look at a wide range of these animals comparing the X and the Y chromosomes to see about how their lifespans stacked up and what happened. And dun-dun-dun! Yes, indeed. It appears that this unguarded X hypothesis is still holding water fairly well. It is a good hypothesis. They have shown that across mammalian species, the women live longer than the men. The double X live longer than the XY. And I'm sorry, Ethan, to bring you this bad news of XY chromosomal issues.
You know, if having a Y chromosome is the worst thing that happens to me in the genetic lottery that we all play, where you're like, oh, thanks for my messed up teeth, Grandpa. Thanks for the male pattern baldness, everyone. Thanks for all this. Like, oh, yeah, well, I got this unguarded X chromosome, and my Y chromosome is full of palindromes because it knows there's only one of them. So, well, at least we'll try and get two chances to get it right. Like, yeah, you know, we're doing the best we can with what we got to work with. And this is what I got to work with. I'm doing all right.
Do it.
Probably not as good as if I had two X's, but I'll take the hit. After all, I'm just looking for my placenta world over here.
So I have a question. Outside of XXXY sexual inheritance, we also have ZW. So ZW is where the female of the species is heterozygote and the male of the species is homozygote. So he's ZZ. Yes. And you see that in reptiles, some birds. There's a very large snake in the Amazon that they discovered that she was heterozygous because she made a bunch of clones of herself. She was able to self-breed. And all of the snake babies were genetically identical to her. So I wonder
Amazing.
what that sort of life expectancy is like.
They looked into this as well. So you are giving me the next step in my story here, which is perfect. Yes, they looked at animals, birds, butterflies, moths, a wide array of ZZ and ZW chromosomes, and they found the same trend occurred. So in those species, the homozygous Zs, the ZZs, which are the males, lived longer than the females, which are ZW. So it's so the reason they have such a higher amount of certainty in the hypothesis now is because they've done this comparison with this natural experiment from these different chromosomal types.
So basically, you have to go to the parent that has the two chromosomes, the double chromosome, the no unguarded chromosome, and when they make you, if you're the heterozygote or the hetero one, you have to hope you get the good one and not the bad one.
Yes, you have to hope that you get the gamete, the X chromosome that has the fewest bad mutations.
All right. So you hear that? Everyone, every male out there listening with a little brother, that's the next thing to tease them about. Tease your little brother. You got the good X chromosome. He got the bad X chromosome. I'm sure of it.
One thing that came out of this study that was incredibly interesting is they expected to see similar life expectancies for the longer lived sex in their study, regardless of whether it was ZZ or XX. And what they found, though, is that the males that are ZZ, they did not live as long as the XX females. So there's something that's still killing the males sooner.
I have theories. I have so many thoughts right now.
Do tell.
Oh man, I would love to know if there's hormonal imbalance or if is there aggression? Are ZZ males more aggressive, and so you're seeing a shortened life expectancy because of behavioral attributes and not genetic attributes?
Right.
Like many questions.
I think the researchers now have new questions as well, and will probably be doing future studies that actually look into those differences very specifically to see what it is that's going on. Because beyond just the homogamete, heterogamete difference, there is something else happening in males that's leading to lower lifespans. But in human males and other mammalian males, it's pretty much that Y chromosome.
How absolutely cool. X chromosome inactivation is kind of incomplete. So yeah, you're right that we can get some of that coverage as XX if something is not functioning entirely right. XY, you're kind of out of luck. Sorry.
Sorry, not sorry? I don't know what to say here.
That incomplete X chromosome inactivation is why all calico cats are female. Right. Yeah, because you can get that sort of mosaic color expression.
Oh, that's interesting. I always forget about that one. All
That's my favorite example of X chromosome inactivation. So I remember it. I'm a biologist because I have, I'm a biologist and I'm a reproductive biologist because I have a favorite example of X chromosome incomplete inactivation. It's a good time.
Yeah. I have a favorite kind of sushi. No. Oh, all right. Ethan, do you have another story to tell us?
I do. So I figured if we were talking about the most energetic bang or explosion that we found in the universe, we should also tell a story of the most energetic explosion since the Big Bang that we know must have happened, but that we haven't found yet. because we have something that's even more energetic, that gives off even more energy than when these supermassive black holes accrete matter, give off these relativistic jets, and blow out these billion light year cavities in the intercluster medium. What
Right.
could that be? Well, we talked earlier about how you get black holes that spiral in and merge. And LIGO, over the last 5 years, has started finding these directly for the first time. It hasn't been officially released yet, but since LIGO started its new run in April of last year, if you add up all the merging black holes that it's found, it's got like 50 of them now. And in every case, we see the same approximate amount of energy get released in the final stages of these mergers, the very final stages where they in spiral, they cross the event horizon of each other, they merge together, and these ripples in space-time, these gravitational waves get emitted with peak amplitude. About 10% of the mass of the smaller black hole of the two that merged, about 10% gets converted into pure energy. So you can start asking, hey, how does that work if your black holes aren't these tiny stellar mass black holes? What if you go up and you consider when these massive galaxies with two black holes at the center of each merge together? What happens then? Guess what? The physics is the same. Einstein's equations give you the same prediction, and it's about 10% of the mass of the smaller black hole that's going to get converted into pure energy. So when you do the calculation and you go from two black holes to one black hole, you can say, you know what? If there's a pair of black holes where the less massive one is more than about 500 million solar masses, that's going to produce more energy than even this giant cavity we found in the center of this galaxy cluster. That's going to produce that energy not on the time scale of millions of years, but we're talking something like weeks or maybe months for the biggest black holes of all. And if you ask, well, how big is the biggest, most massive black hole we've ever found? You're talking in the tens of billions. The single most massive black hole we found is some 60 billion solar masses. So when you say, what's the most massive, most energetic energy output event in the whole universe throughout its history, it's got to be from these mergers of these supermassive black holes, these ultra supermassive black holes. What's fascinating is we have a type of detector operating today that could detect them. You have to get lucky because these are rare events. There are, like I said, about 2 trillion galaxies in the universe, but only a fraction of a percent of them, only a fraction of 1% of those galaxies have these mega billion solar mass black holes, but they merge together. We find, if you look at like the Coma cluster, there are 2 giant elliptical galaxies that are bigger than any other galaxy in there. One of them has a black hole that's like 20 billion solar masses. The other one has an unknown mass black hole. Someday they will merge together. When they merge together, they're going to produce more energy and gravitational waves than anything else we've ever seen in the universe. And that's just in our own backyard. We look out in the universe, we see these supermassive black hole pairs getting ready to merge together in these massive galaxies. We see examples where we know supermassive black holes merged in the past. What's happening with these gravitational waves is they come towards us, and these are what we call low frequency black holes. Frequency is how often do these black holes spin around before they merge together. And what LIGO can see is black holes that orbit each other a few times a second or a few dozen times a second or maybe even 100 times a second. These black holes are so big and their event horizons are so large that we're talking they're going to be orbiting each other with a period of like months, years when they get ready to collide. So when this happens, they're going to create these enormous, long period ripples in space. You can't see them with a gravitational wave detector like LIGO because you can't build it big enough. You'd have to build a gravitational wave detector that went from like the sun to Jupiter to be able to see it. We're not going to build one of those anytime soon. Too bad. So what do you do instead? You go and take a look at the best natural clocks in the universe, pulsars. You look at a pulsar here and there and there. You look at these pulsars all over the place, and you start measuring their timing and syncing them up. And when these enormous ripples in space pass through them, they're going to perturb the pulsar orbits together. So you take all these accurate pulsars in all these different directions, you start monitoring them. And that's what the Nanograv collaboration in North America and the Parkes Pulsar Timing Array and the European Pulsar Timing Array, they're all pooling their data together to observe all these pulsars and say, can I detect these ripples? According to the scientists working on that, they are expecting that at some point over the next decade, if they've calculated the event rates of these things right, they will break the record. They will find evidence of the most energetic merging black holes we've ever seen. So this is not something we're 100% sure we're going to find, but we are 100% sure these events exist in our universe, and it should only be a matter of time and technology before we find them. And to me, that's incredible. There's a cosmic record out there that we know is just waiting to be broken, and it's up to us to go and track it down, and we're on the case. And that's amazing to me.
I think it's so the whole use of pulsars to be able to create the basically a LIGO without having to build it. I mean, it's awesome. It's just amazing. It's like, oh, we have these things. Their timing. The timing is this way, and we can match them up this way. And because we know their distance and blah, 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 all this stuff. Now let's look for these little signals.
You know, I don't get a chance to nerd out about pulsars very often, so I will tell you some of the coolest facts I know about them. First off, the fastest one rotates 766 times a second, which means that's how many ticks, tick, 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 every second we get. Second, up until atomic clocks improved past a certain point, pulsars were the most accurate clocks in the universe, and pulsar timing is still getting better. It is possible, although not likely because atomic clocks are getting better too, it is possible that pulsars will one day become the record holders again. Third off, pulsars are so accurate that you can start watching one tick, 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 tick. You can look away for like a year. And you can look back after a year and know whether 3 billion pulses have passed or whether it's been 3,000,000,001 pulses that have passed. They are that accurate in timing it. And that is why we can get such good data out of them, is because these are super regular events in the universe. Very rarely a pulsar will do something called glitch. That happens when stars have a starquake, right? So you know that Earth has earthquakes, and what that is, if you're a geologist, is that is the moment of inertia of Earth, the way the mass is distributed inside, rearranging itself. When we had that big earthquake in Japan a few years ago, it rearranged the moment of inertia of Earth so much that our rate of rotation changed by like 2 microseconds. So when
stuff like that happens in a star,
what happens? That's absolutely right. The pulsar timing changes. It glitches. We saw one of those happen back in the 1980s. They're very rare, but when one of them happens, that's the only thing that can screw up this millisecond pulsar timing method.
Yeah, little star burps. Let's not have no star burps. You're going to mess up our timing. Glitch.
Star burp.
Yeah. But I don't know. Maybe we shouldn't be calling them glitches anymore. Maybe we should go with Princess Vanellope. I saw that movie. It was pretty good.
Oh, yeah. Oh, she's still good. Still good.
I didn't see it. I'm sorry.
Oh, Wreck-It Ralph?
You got it. You got it.
Hide me to the internet, Dr. Kiki.
And
watch Wreck-It Ralph and Ralph Breaks the Internet, which is so darling. You have homework.
Okay, I will get on it. I like homework like this. It's my favorite kind.
Good.
Bring it. All right, we have merging super massive black holes, pulsars. We've got all sorts of fun. And Jessica, what fun do you have?
Back to the biology and to our second sort of placenta story. BPA-free does not mean safety. Using BPA-free plastic products could be as harmful to human health as using BPA-containing plastics, including a developing brain, as those products contain the controversial chemical known as bisphenol S. And this is from a new study led by the University of Missouri and published in the Proceedings of the National Academy of Sciences. For decades, scientists have studied BPA extensively in animal models, with results indicating that the chemical plays a role in early pregnancy loss, placental diseases, and various negative health outcomes after birth. But these adverse health outcomes have become more widely known, and so companies turn to BPA alternatives and often label them BPA-free. So you'll see Tupperware or plastics out in the grocery store with a little BPA-free tag. It turns out that one of those chemical alternatives, bisphenol S, is still not safe for people to use. So in the study, Rosenfeld and her colleagues focused on examining the effects of BPS on a mouse's placenta. And she said that the placenta serves as a historical record of what the unborn child faces while in the womb. And the placenta can transfer what mother gives it to the fetus. And synthetic chemicals like BPS can penetrate through the maternal placenta. So whatever is circulating in mom's blood can easily be transferred to the developing child. The mouse model is an excellent model for understanding this because a mouse placenta is very similar to a human placenta in terms of cardiovascular structure and being able to deliver blood and nutrients through to the baby. So they have a lot of similarities. Rosenfeld's research is a piece in translational medicine and has found that developmental exposure to BPA or even its substitute BPS can lead to long-standing health consequences. So BPA-free, maybe not for me.
You got some nice rhymes in here.
Thanks. That's why I get paid small amounts of money to leave places quietly.
Yeah, you're like a BPS poet. Bips poet. So they're looking at a mouse. They're not looking at human placentas. There's got to be lots of differences between. We talk a lot on this show about, hey, we cured this in mice. We discovered this in mice, and so often it's very different from what's actually happening in people. Even though we use mice as this model species, like what are? Do you know how similar are these mouse placenta, and how much can we really take from? Or what the implications from a study like this? Because I'm tight.
I do know how similar a mouse and human placenta are. And in terms of delivery from mom to baby, they're very, very similar. So you have two sides of the placenta. You have the mom side and you have the baby side. And in mice, just like in humans, in the uterine lining, you slow blood flow down as it goes from mom to baby. When it hits the sort of middle of the placenta in a mouse, it's called a labyrinth, and that's where the nutrient exchange takes place. It's not where David Bowie lives. Mm-hmm. And in humans, you have this chorionic plate that surrounds the syncytia trophoblast, and that's where the exchange of nutrients takes place in a human placenta. So the mechanism of exchange is very similar, but the actual physical mechanics are a little bit different in between them. Right. But they're both discoid placentas, so they're similarly shaped. They
kind of like the Milky Way galaxy?
Yeah. And fun, so discoid, and the more oblong you get your placentas, whether you're a mouse or a human, the worse it actually is. You want a nice round placenta. Oblong placentas are more of a sign of fetal malnutrition, like the placenta has tried to grow further out in order to spread itself out a little bit. So it's a sign that there might be some sort of fetal growth restriction going on. So mouse and placenta, human placenta, definitely close enough to call this a good model organism for this particular study.
People think BPA is bad, but I was born in the 70s. I know my mom drank when she was pregnant with me, and look how I turned out. No, don't look how I turned out. So how bad is BPA really? Or how bad is these BPA-free substitutes that have whatever crazy new chemicals they have instead in them? Are they as bad as, drinking enough alcohol that they'll tell you, but not enough alcohol to give you fetal alcohol syndrome?
And there was a study that just came out again about how much alcohol is safe in pregnancy, like within the last 2 weeks. And the answer on that study was absolutely 0. It is not safe to tell a mom that she's allowed to drink. So
okay,
we're back on that bandwagon.
So whatever dice roll happened there, don't do it. Don't roll those dice, kids.
I mean, there's what causes fetal alcohol syndrome? Drinking alcohol while you're pregnant.
Is it alcohol? I would guess.
It is.
I would guess.
It's taking the Everclear you had left over from making your antibacterial, your hand sanitizer, and chugging it instead while you're pregnant. Like, maybe don't do that.
Yeah, but you don't want the baby to get sick, so you have to kill the germs inside you. And what better way is there to do that than with Everclear? Are you going to just use your immune system? What?
Ethan, that is not how science works.
So you are just going to use your immune system then?
I'm young and healthy. I don't know. Like you said you were born in the 70s. Old man, like you're going to die soon. You better be careful with the COVID.
You think so with my unguarded X chromosome. It's all coming down now.
Unguarded boomer chromosome.
The haunted death from the biologist. It's because of that placenta pun, isn't it?
Yeah. So
don't touch your nose.
I do what I want.
The bad stuff from the BPS, the BPA, like what are the health detriments over the long term?
The concern is endocrine disruption. And endocrine disruption means that you're going to produce more of particularly estrogen, but other hormones in your body than you really need. And that has detrimental effects for longevity, cellular functionality. And you can actually get what they're calling like I don't really like the word. I hope that they come up with something else, but a genderization of organs. Yeah. So the liver definitely can be more female or more male, depending on what chemicals are being released by your hypothalamus. And the hypothalamus can be affected by BPA and BPS. So yeah, you don't want to mess up your organs with these endocrine disruptors, and they're saying
mess up my hypothalamus. Jeez.
Yeah, so BPA definitely affects the hypothalamus, and it accumulates in the mitochondria at a faster rate than it accumulates anywhere else. And there are researchers actually working on BPA environmental exposure and long-term health at OHSU, so it's a really neat thing to be looking at right now. So the study came up and I thought it was pretty neat. So look at PNAS. Again, that research came to us from the University of Missouri, all about BPA products not being safe.
Not being safe. It's not just BPA, but BPA-free
BPA-free, yes.
If they're using an alternative that's still related to it, it's still not good. These alternatives are not necessarily good.
I'll just stick to using my lead water bottle. That's the way to go.
Ooh,
there you go.
Bad news.
Ooch, not great. Hey, everyone. This is This Week in Science. Do you want to help TWIS grow? I know you do. Tell a friend today to subscribe. Go to twis.org. All right. This is the time for the quick stories to jump in and jump out and have a little bit of conversation, but not too much because these are quick stories and not long stories. So I just want to tell you very quickly, our early Earth may have been a total water world. It might have not really had continents the way that we know them. Researchers at the Colorado State University in Boulder have been looking at oxygen isotopes in really old rocks in Australia. I mean, pretty much, You can go to different places on the planet. Australia is one of those places that has really, really old rocks. So they found some rocks that are 3.2 billion years old. It's a chunk of old oceanic crust. Got flipped on its side a little bit. And they started looking at, like I said, oxygen isotopes. They were looking at the ratio between oxygen 18, which is heavier isotope, and oxygen 16, which is a lighter isotope. They found that the ratio of the isotopes was different 3.2 billion years ago, so that there was more of the heavy oxygen 18 than there is now in the seawater. And because they figured that out, what they think happened, they're looking at today's land masses and the ratio of oxygen 16 to 18 in land masses today versus the stuff that was under the ocean for a very long time. And based on the differences that they're seeing, they're saying that something happened a long time ago to start shifting the chemistry of the ocean, but also that they think a large part, if not all, of our planet was once covered in water. But at least now we have a way to test for it. And the final take-home message here is that it wasn't just, oh, look, the planet, and then there were continents, and that's the way it was. There may be kind of different, there may be a couple of timelines that we can look at and explore. A different timeline for our planet's history.
Yeah, I took a look at that study and I thought it was fascinating because they have a couple of different scenarios that this could have happened, like that could have caused this. One of them is pretty simple and straightforward and doesn't involve any weird stuff with a total water world. It's just, hey, what if the oceans were warmer? Then that could have changed the oxygen 18, oxygen 16 isotope ratio. But the other one is, well, what if the chemistry was different? What if the chemistry, because the environment was different between the water and the landmass that was interacting? And that could have been in a different chemical environment, which would have happened totally underwater. If that's what happened, that's going to be very interesting, because then when we talk about geology and atmospheric science, we have a problem now on Earth, which is how do you get rid of all of that water? How do you get rid of all of that extra water that would have been here
covering everything
3+ billion years ago? Because if you take a look at how much land do we have, how much ocean do we have, what's the topography of Earth, there's no way we would have had a total 100% water world with only the amount of ocean we have on Earth today. So it had to have gone somewhere. We don't know the answer, but this is one of the neat steps in like science in progress.
Yeah, it's a cool question. What did happen? I think the Earth ate it.
Polar ice caps, anyone? Or
suck it up and pull it down. Maybe it had acid indigestion at its core.
Or how much water was in our atmosphere in early Earth, if maybe that changed.
Maybe that's the difference as well. Mm-hmm. Yeah. We don't know. Mysteries, more mysteries and questions.
I love science.
No. Okay, Ethan, what's your last quick story?
My last quick story, March 19th, everyone in the United States, are you ready? March 19th is the equinox. No matter where you are in the United States, March 19th this year is the equinox. Does that sound like a weird sentence to you? For those of you who remember living in the 20th century, it sure should. Equinox is always on the 20th or 21st. What's it doing on the 19th?
Yeah.
The answer is this is the earliest equinox in 124 years, the earliest equinox since 1896. What happened? Why is the equinox so early this year? The answer is it's been slowly getting earlier because we haven't been correcting the calendar for leap years constantly. We do it all in chunks. So 1700 wasn't a leap year because it's not a year that's divisible by 400. And 1800 wasn't a leap year. And 1900 wasn't a leap year. Every fourth year on the 100 years where you don't get a leap year, guess what happens? That helps push the equinox to a later date. but we messed up. And we didn't mess up. This is perfect timekeeping. But 2000 was a leap year because we keep track of leap years. They're every 4 years, except on the century, unless that century is also divisible by 400. And 2000 was, which means ever since 1900, ever since that passed, every 4th year where we have a leap year, that equinox gets earlier. So 1904's equinox was about 45 minutes earlier than 1900's equinox. And as we go every 4 years, we've been getting 45 minutes earlier. Guess what? This is the first year since 1896 where it's March 19th everywhere in the United States. Guess what? Every 4 years, that's still going to get worse. When we get to 2096, it's going to be equinox March 19th in the morning everywhere in the United States. It's going to be March 19th for your equinox everywhere in Europe, everywhere in Africa, and even over large parts of Asia as far east as New Delhi. So enjoy having your early equinoxes. They're going to keep getting earlier every 4 years. And if you're like, oh, I want my equinox to be on March 21st again, how long do I have to wait? Your answer is 2103. So hang on if you can. 2103 equinox goes back to March 21st, like I remember when I was a kid.
How does it reset? If it's
because 2100 isn't a leap year.
It's not a leap.
So we get 8 years or 7 years in a row with no leap years, and that bumps the calendar back so that your equinox comes later.
Got it. Resetting the clock a bit. This is great. Calendar math. Astronomical calendar math. I
know. Well, you got to keep track of your sidereal years, or they all,
because our sense of time doesn't quite match up with what our planet is actually doing in orbit around the sun.
Hey, at least 3/4 of your listeners know that the planet is round and revolving. Hopefully we'll get that last quarter, too. You guys are in on this, right? Yes. All right, good.
They better. All right, Jessica, tell me some CRISPR news.
Yeah, this is a bleeding edge story that just came out this morning.
Beep, 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 beep. Sorry.
For the first time, scientists have used the gene editing technique CRISPR to try to edit a gene while the DNA is still inside a person's body. The groundbreaking procedure involved injecting the microscopic gene editing tool into the eye of a patient blinded by a rare genetic disorder in hopes of enabling the volunteer to see. They hope to know within weeks whether the approach is working, and if so, to know within 2 to 3 months how much vision will be restored. In this new experiment, doctors at the Casey Eye Institute at OHSU in Portland, Oregon, yay, my home institution, they injected it into the eye of a patient who is nearly blind from a condition called Leber congenital amaurosis. So they injected microscopic droplets carrying a harmless virus that had been engineered to deliver CRISPR into the editing machinery. Beginning in infancy, this rare genetic condition progressively destroys light-sensing cells in the retina that are necessary for vision. Vision impairment varies widely, but most patients are legally blind and are only able to differentiate between light and dark, or perhaps to detect movement. So the goal is that once the virus carrying CRISPR has been infused into the eye, then the gene editing tool can slice out the genetic defect that caused the blindness and maybe hopefully restore some of the protein that and prevent death of cells in the retina and revive other cells. So CRISPR directly in somebody's eye, and they say it's the first time it's been used, by researchers. We know that biohackers will sometimes stick their butts full of a needle full of CRISPR.
It's true.
But this is the first time it's been used by people who knew what they were doing. To hopefully fix a really terrible condition. So there's hope.
That's cool. It'll be really interesting to see what happens there. I wonder if it's the kind of thing also, do you get one shot in the eye one time, or is this the kind of thing where you're like, I think I have to go back a few times?
Right. We don't know what the degradation is like or how effective it's going to be. So this isn't even a paper yet. This is hey, by the way, we just did this and we want you to know, and we'll We'll see how it goes. This is very different than putting CRISPR in gametes. These are non-gametic cells. So there are fewer ethical concerns about it. And we hope that this person is able to regain some of their sight. It would be very exciting.
And it's being done in a medically approved clinical situation, not some fly-by-night closet. So don't go out looking for stem cell or CRISPR injections. Wait for the science to come out, everybody. It's very important. Very important. Thank you. I think we've come to the end of the show.
Oh,
what?
I know. What? It always goes by, and then I'm like, no.
Where we miss Justin and Blair, we will happily come back anytime that they happen to be off traipsing the world like Indiana Jones.
That's right.
Yeah.
Yeah, I would love to have you both back again. This has been so much fun. Thank you so much for joining me tonight.
Oh, it's been our pleasure. I'm sure we'll make it happen again soon, either separately or together again, where Jessica can make fun of my unguarded X chromosomes some more, along with the male pattern baldness and whatever else is the topic du jour.
I'm sorry, Ethan.
We'll all cheers with the Everclear.
Now that's a jet.
When you're a jet, you're a jet.
Thank you for having me, and thank you so much, TWIS audience. You were the absolute best. It's been a lot of fun.
Where can people find you if they would like to follow you away from This Week in Science? Jessica.
I'll go first, sure.
Or Ethan, go. Yes.
Okay. I'm Starts With a Bang on Twitter. I run the blog Starts With a Bang on Forbes.com. I have a Patreon. I do podcasts. I'm the Starts With a Bang podcast on SoundCloud and in the iTunes store. And if you are looking for me, my name's Ethan Seigel. Starts With a Bang. Find it. Also, check out my first 2 books, Beyond the Galaxy and Treknology. You'll be glad you did.
You can find me on Twitter. I'm Dame_DNA, and I talk about all things placenta at #Team Placenta on Twitter. I'm on Instagram as Dr.Jessica Hebert, and I also post on Scientists Who Selfie. And if you're into science-based music, please check out my Portland-based nerd trio, the PDX Broadsides. We just came back from our first East Coast tour. We released our 5th album, Relatable Content, last summer. You can find all of that music, the PDX Broadsides, on Spotify, Twitter, and where other mediocre music is sold.
They're TWIS tested and TWIS approved.
It's true.
It is true. Yes. All right. I would also love to shout out some thanks to Fada for his help in the chat room and also with social media and show notes. Thank you so much for your help. Gord McLeod for manning our main chat room, Identity4 for recording the show, and I would love to thank our Patreon sponsors and the Burroughs Welcome Fund for their generous support. Thank you to Paul Disney, Ed Dyer, Stu Pollock, Andrew Swanson, Craig Landon, Ed, Philip Shane, Ken Hayes, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Richard Porter, Mark Mazzaros, Bob Calder, Jack, Matthew Litwin, Jason Roberts, Bill Kay, Eric Knapp, Richard Brian Condren, Dave Neighbor, Ashley Doyle, Maddie Perrin, Ben Bignell, Justin Taylor, P.S. Josiah Zahner, Howard Tan, Donald Mundis, Sarah Forfar, Dan K., Matt Bass, Darwin Hannon, Patrick Pecoraro, Jean Tellier, John Gridley, Corinne Benton, Adam LaJoy, Sarah Chavis, Rodney Lewis, Tiffany Boyd, Mountain Sloth, Seth O'Gradney, Stephen Albaran, John Ratnaswamy, Dave Friedel, Daryl Myshak, Paul Ronovich, Sue Doster, Dave Wilkinson, Noodles, Kevin Reardon, Christoph Zucknarek, Ashish Pants, Ulysses Adkins, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Suzuki, Jim Trapeau, Craig Riley, Sean Lamb, Steve Leisman, Kurt Larson, Rudy Garcia, Marjorie, Gary S., Robert, Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Burke Hessenflow, Kevin Parichin, Byron Lee, and E.O. Thank you for your support on Patreon. And if you are interested in supporting us, you can find information at patreon.com/thisweekinscience. On next week's show,
we will be back on Wednesday at 8 p.m. Pacific Time, broadcasting live from our YouTube and Facebook channels and from twis.org/live.
Want to listen to TWIS as a podcast? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe too.
And for more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twis.org. And you can also sign up for our newsletter.
You can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just put TWIS in the subject line so your email doesn't get spam filtered into oblivion.
And you can also ping us on Twitter, where we are at TWI Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We love your feedback. If there is a topic you would like us to cover or address, or a suggestion for an interview, please let us know.
Dr. Kiki and Justin and Blair will be back here next week, and we hope that you'll join us again for more great science news.
And if you've learned anything from the show, remember...
It's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say, and if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.